Right. I'm back to finish this discussion on genetics off with you guys, and I want to talk about some chromosomal problems here. So as we know, hopefully now as humans, that we have 23 pairs of chromosomes, 23 homologous pairs, and that makes 46 total. Pairs 1 through 22 are referred to as the autosomes, and the 23rd pair are the sex chromosomes. So I think probably by now you have a pretty good understanding of the fact that the number of copies of each gene that we have is very important. So when we start to think about chromosomal problems, there's something going on with the genes that has deleterious consequences in most cases. So the first example is one that we do not see in humans. It's not viable in humans, but we do see in some plants, and that example is called polyploidy. So essentially with polyploidy, we have one or more complete sets of extra chromosomes. So in the example of some of these flowering plants, we see two sperm fertilizing the same egg. That would be an example of polyploidy. I'm not going to spend a bunch of time talking about it because it's not something we see in humans. However, aneuploidy, it's a hard word to say, aneuploidy is something that we do sometimes see in humans, and that is either extra or missing chromosomes. So really what aneuploidy comes down to is it's an error of meiosis. And it can happen in either the first or the second meiotic division, but essentially what's going to happen is the chromosomes aren't going to separate correctly. So if it happens in the first division, meiotic division, instead of one of the chromosomes of the pair going to one cell and the other going to the other cell, both would go to one cell and neither would go to the other. So that's one type of non-disjunction that happens in the first meiotic division. It can happen again at the second meiotic division in which the sisters get pulled apart. So essentially in one of our gametes we'd have two sisters, and in the other one we'd have none. So let me show you a picture of this. And before I show you a picture, so there's lots of different opportunities for this to happen, and it can happen on any chromosome at, either in the first or the second division cycle. But in many cases, and probably in most cases, I would venture to say, this is going to be a game over kind of a deal. So you don't even know that it's happened. It probably will happen so quickly. A miscarriage would occur before the pregnancy was even detected. However, there are cases where it's not quite as catastrophic, and so I'll talk about some of those. So here's just a picture of what this non-disjunction is all about. So non-disjunction and aneuploidy are pretty much the same thing, or they are the same thing. So this is normal over here. So just check it out. So here we've got, and just to keep it easy, we're just keeping track of a couple chromosomes. So here's one homologous pair, and then during meiosis 1, this pair would ultimately separate, right? And so we'd have one of the pairs goes here, one of the pairs goes here, and then we're going to divide them again, and the sisters are going to get pulled apart. So these would be our gametes, right? So normally we would have 23 chromatids in each of these cells in a human. So let's flip to here. So here's where we have non-disjunction in meiosis 1. So instead of one of these chromosomes going to each cell, both of them went into one. So this one has extra and this has none of that particular chromosome. So then if this didn't, exit out right at that moment, which in most cases it would, what we'd have is ultimately our gametes, none of them would be right? They're all screwed up because these guys are going to separate. And so so remember, there's only supposed to be one of these in each. So you have two of them, 24 chromatids, and this one 24 chromatids, and this one 22 here and 22 here. So none of these are viable. Now, this one is where we see non-disjunction in meiosis 2. So everything's cool here, still good, But when these guys separate, that's where the problem is. So these guys separate okay. So 50% of the gametes are okay, but the other 50% are abnormal. This one has none, so it'd be 22 chromatids, and this one has 24. So that's an extra, and that's -1. So those are examples of non-disjunction, just in a sort of a theoretical sense. Let me give you an example here of non-disjunction or aneuploidy in a human. That's pretty common. And so just take a look at this. This is a karyotype. We've seen these before. So we've got, we're just looking for something to be weird. And so this is number 1. Those look cool. 2 of them. That's what you're supposed to have. They're the same size. It's sort of hard to tell because this one's bent, but if you were to straighten it up, they'd be the same. Number 2 is good. Number 3, good, 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 good. Everything's good. 13, 14, 15, 16, 17, 18, 19, cool. 20, good. Uh-oh. Here we get to 21. There's 3. There's only supposed to be 2. So we've got 3 number 21s, which is also called trisomy 21. This would also be a female. You see there's 2 X's and no Y. So this would be a female with Down syndrome. Trisomy 21 is Down syndrome. So it is called trisomy 21. Tri for 3. Chromosome 21 is the chromosome where there's the problem, and it is the most common cause of Down syndrome. We don't really know what causes it. We think non-disjunction might increase with age. I think that there might be more to it than that. We're seeing more and more Down syndrome with younger females as well. So I think there are definitely some environmental factors at play here. But what we do know is that it's pretty common in the scheme of polyploidy, aneuploidy. There's also a trisomy 18. There's a trisomy 13 are the next most common, but trisomy 21 is the most common. And there's probably others, but we never know about them because the error is incompatible with life. So here's a couple other examples. So this first one would be, these are extra or missing specifically sex chromosomes. The first is XXX. So XXX is where a woman has 3 X chromosomes instead of 2. It happens in about 1 every 1,000 female births. Doesn't really seem to be that big of a deal. With XXX, women have female sex characteristics and they're fertile, which means they're able to have children. There may be some associations with learning difficulties, learning problems, maybe some slow to develop motor skills in infants, and there might be some trouble with muscle tone. The next one on our list is Klinefelter syndrome, which is XXY. So here we have a male XY with an extra X. So it's possible to have even more X's, which would be like XXXY or XXXXY. So this is called Klinefelter's syndrome. It's not that uncommon. Actually, that number on there is not the number that I have in my notes written down. I have Klinefelter's thought to affect one out of every 5 to 1,000 male births. I think that number is probably needs to be adjusted on my slide. So Klinefelter's syndrome, the symptoms are pretty variable. Men tend to be infertile, and they have kind of like less strong male sex characteristics, so their body is not quite as dense, and they tend to not have as much facial hair as other men. But that's the biggest deal with Klinefelter's syndrome. I'm going to talk in a little bit in the next slide about X inactivation, which is very common in women. So when women have 2 X chromosomes, and they don't need 2, right? Though all the pertinent information, same information is found on both. So we inactivate one of them, and we females, I should say, one of them is inactivated. So it's the same thing happens with Klinefelter syndrome. One of those X's gets inactivated. The next one on our list is XYY, which is a male with an extra Y. And this is in about one in every 1,000 births. And it's very similar, at least from my investigation. I'm not an expert on this by any stretch, but it's really similar to the triple X in females in that it doesn't seem to be that big of a deal. The males are fertile, so they're able to reproduce. They appear to be taller than the average male, and they have the same kind of learning disabilities and maybe like poor muscle tone and late motor skill development in the infants. But it doesn't seem to be that big of a deal either. And then we have Turner's syndrome. Turner's syndrome is XO, so it's basically a woman that lacks part or one of her X chromosomes. So she has just one functional X. So people with Turner's develop as females, but oftentimes they have a short stature, so they're shorter. They most likely are going to be infertile, and they have some learning difficulties as well. Turner's syndrome, I think that's overestimated as well on our slide. I have one in every 2,500 female births. But it's not that big of a deal because, like I just said, one of the X is inactivated anyway. So you see they're infertile, they're short of stature, they might have some heart problems. The webbed neck means that their neck kind of it's more triangular shaped at the base, so it's as you get to where your shoulders, at the bottom of your neck and the top of your shoulders are, it kind of goes out more as an angle. They might be prone to some endocrine abnormalities, including diabetes. It seems like a lot of the fetuses don't develop, which you can see there. But if they do develop and are born healthy, that doesn't seem to really be that big of a deal. And then finally, we have just a Y with no X, and that doesn't happen because it's incompatible with life. There's too many important chromosomes on the X, or genes, on the X to survive that. So that's something that's not compatible with life. So those are some examples of sex chromosome abnormalities. So I've been talking about this a little bit, and it's the deal with females having 2 copies of genes on the X chromosome and men only have 1. And that's, like I said, it's not that big of a deal because it seems like 1 is redundant, right? We only need the 1. All the pertinent information is on 1. So normally if you have 2 copies of a gene, that would be bad. But in this case, it's not that big of a deal because females inactivate the X. Which X gets inactivated is random in humans. And in most animals, with the exception of kangaroos. And kangaroos, only the parental X is the one, always the parental X is the one that gets, so the one that comes from the one that comes from the father is the one that gets inactivated. But in human females, it's random which one gets inactivated. And it's kind of complex how it happens, but essentially there's a cell that shuts off all but one of X chromosomes in each cell. So one of them kind of goes dormant. And when it goes dormant, it gets a name, and that is called a BAR, B-A-R-R body. So it sort of coils up into sort of like a ball, and it's not functional. There is kind of an interesting, what am I trying to say? Trivia game show question kind of a thing with these calico cats. And calico and tortoiseshell cats are an example of X inactivation. So basically, if a female cat is a heterozygote for both the black and tan alleles for the coat color gene. The coat color gene is found on the X chromosome in cats. So she'll inactivate her X's and randomly and at different times during development. And the result would be alternating black and tan or black and orange fur, depending on which X's have been inactivated. And then, during development, and then in some cells, one of the X's, the X for the black coat color has been inactivated. And in other cells, the X that carries the orange coat color gene has been inactivated. And then those cells continue to divide and divide and divide, just like what happens during the process of development. So the resulting cat will have patches of black and orange fur, or black and tan fur in the case of the tortoiseshell cat. So those tortoiseshell cats and calico cats are almost always female, and that's a result of this inactivation, these patches of fur. So, and that's perfectly normal. Like, it's a normal development. It's nothing weird about that. And then finally, there's some smaller scale abnormalities that are worth noting. One is if we have a deletion of one or more genes. And there's an example of a condition called cri du cat, or cri du chat in French, which is a deletion of several genes on the X chromosome, or on the 5th chromosome, pardon me, and it results in what sounds like a screaming or squealing cat when the baby screams, and it's a chromosomal abnormality. Every once in a while, we'll have a duplication where multiple copies of one or more genes is produced, and that's probably actually plays an important role in mutation, because we have this kind of like spare gene. And then finally we have something called an inversion, where non-homologous chromosomes exchange parts, and that's kind of weird, and it sort of depends on what genes are exchanging parts. But there's a type of leukemia called chronic myelogenous leukemia, which has which in 95% of the people who have that particular type of leukemia, they have a translocation that causes a protein to speed up cell division and suppress cell death. And that is it is a consequence of inversion, where we've switched non-homologous chromosomes have exchanged parts. So those are some examples of smaller scale abnormalities. So we're like, it's more on the gene level versus on the chromosome level: deletion, duplication, and inversion. So in a nutshell, genetics is kind of challenging. There's a lot going on. And it can be a little bit complicated, but what I'd like you to take out of it is just sort of the basics, just sort of the basic understanding of what's happening, what a pedigree is, what all that means. You want to be aware of the questions that you ask when you're looking at a pedigree in terms of how to determine if this is autosomal or sex-linked, and dominant versus recessive. Have a basic understanding of polyploidy, aneuploidy, those kinds of things. So study this a little bit. I'm going to put an activity up for you guys to just sort of test your understanding. And then when you come to class on our next class session, we're going to be doing some in-class work on both cell division and also some genetics problems. So come prepared with your notes and your knowledge of these topics. All right, I will see you then.